So thank you, Lou, and thank you to Chris and the others for the invitation. It's actually a really deep pleasure for me to come here and give a talk. Lou mentioned the courses in Trieste, which is where I first met Tom Hallam and Lou. The experience of teaching in these courses, and in particular the conference that followed one of them, had a huge impact on my subsequent research. I owe you and Tom a lot, and it's great to be here. And what I'm going to do today is talk about a contribution that mathematical models can make to a much larger study on an emerging challenge, which is understanding the environmental implications of engineered nanomaterials. And like Lou said in the introduction, I'm doing this in the context of being part of a large group of individuals in the UC Center for Environmental Implications of Nanotechnology, funded by EPA and NSF. And I'm going to talk about work which actually involves collaboration with a huge number of people. I've listed some of the closest ones here, and the people whose names are underlined are specifically featured in the talk. I'll try to mention them as we go along, but if not, I want to acknowledge them now. And I think the take-home message I hope from the presentation will be that mathematical models can make modest but significant contributions to a much larger exercise. There's going to be no grand take-home message, no great breakthrough achievement here. It's a succession of incremental advances where modeling is one of a large number of approaches. But it's relevant to a center like this because I'm going to emphasize the role of generic models, the kind of models that many people think are oversimplified, or some biologists think are oversimplified, in synthesizing a lot of information. And I think there is a need for an unarguable need for synthesis in ecotoxicology. And in fact, one of the first talks I ever heard about ecotoxicology was a talk by Tom Hallam at one of the Trieste meetings you talk about. And when you search for papers on the population impacts of population dynamics associated with ecotoxicology, some of Tom's papers are some of the earliest ones that you find. But I think the background, which is fairly basic to the group of us here, is that contaminants impact at all levels of biological organization, individuals, populations, communities, ecosystems. That most, and really all but the most extreme examples of stress through contaminants occur in situations where they're just one of many factors that impact ecological communities. And therefore, we need a way of thinking about them which lets us consider that they act simultaneously with lots of others. And the premise behind this talk is that we need some biology-based or mechanistic theory to help us here, and not just a large number of statistical correlations. And in particular, we can't rely on pure empiricism because of the sheer scale of the problem. The data I've got on this slide came from a talk by Andrew Nell, the director of the CEN, and it was a couple of years ago, so probably they're a little bit out of date. But he quotes things like 75,000 chemicals currently in registered for commercial use in the US. There's probably more. A very small number that have undergone the complete suite of toxicity tests, and the overwhelming cost of a complete suite of tests, especially when they include animal models for any particular chemical. There's no way that even with a quintupling of budget, the problem would be solved with more testing. Nanotechnology has made the challenge tougher. For the purposes of this presentation, I'm simply going to interpret this phrase as relating to the traditional, or the now broadly accepted definition of nanomaterials or nanoparticles. Nanoparticle is a particle with a characteristic dimension between 1 and 100 nanometers, and that possesses properties that are different from either the bulk material or the property or a substance in solution, which is why there is special interest. We all encounter and work with them and are exposed to them regularly. Examples would be metal oxides that feature in many personal care products, notably sunscreen. Titanium dioxide is in probably many things that need to be white. Zinc oxide certainly features in many sunscreens. A number of antibacterial agents. Many of us perhaps have silver nanoparticles in our socks. And then there are a load of electronic applications. I'm going to mention one called quantum dots that is used in photophysics, but there are others. And the relevance of nanomaterials to the presentation to the slide I just showed is that while there may be 75,000 or thereabouts different chemicals in use, nanoparticles for any one substance, let's say zinc oxide, come in many shapes and sizes, and the whole point of designing them is so that they have different chemical properties relating to the shape and size, and because people want to control things like the rate they dissolve or don't dissolve and so on, they commonly come with coatings. When people are interested in say UV absorption, they will dope them with impurities. The range of properties is almost limitless, so we need something that is something that is general. And those of us in the ecology component of the CEN are interested in the effects ultimately on populations and in ecosystem services. And here the concerns are no different from any other area of ecotoxicology. We're interested in potentially effects on energy fluxes, nutrient fluxes, interested in population implications. And a priori, we have reason to believe that there are potential effects, and I just mentioned here one example, the fact that if you specifically design a material for its antibacterial properties and it finishes up in the soil, there is going to be some effect on, or potentially some effect on the soil bacteria. So where do we get data from? And we get data from a range of levels of biological organization that, once you're set up, have very different costs of gathering. As an example, there are many studies that use high throughput, sometimes robotic systems, capable of doing huge numbers of measurements per day of a very stylized form on single cells or groups of cells. Much of this technology developed for biomedical applications. So it's possible to gather a huge amount of information of questionable ecological relevance, but that gives some information on biological process. As you move up the scale, you can perhaps reach the point where with the smallest organisms, like a nematode or a Daphnia, you can do an intermediate number of experiments at a manageable cost, through to the point where complete in vivo studies in, say, a mouse, the sort that as are used in many medical applications, or even more an ecological investigation of what's going on in the stream, are so expensive that you can only do a few. The challenge, as I see it, for theorists is for us to come up with some conceptual framework that lets us get the maximum benefit from the vast amount of partially disconnected information at the lower levels and use it to help answer ecological questions. We're never going to reach the point where we can do experiments on single cells and predict what will happen in a stream. But knowing something about what can happen with single cells can help us constrain the options in a stream and perhaps do better experiments. And that is the broad philosophy that I think underlines the group of people that I work with in this center. I want to argue for an approach that emphasizes mechanism, but before doing so, I want to introduce, in case some people are unfamiliar with it, one of the traditional groups of metrics that are commonly used in ecotoxicology and argue why it has limited value. The standard way of doing of approaching toxicity for many contaminants is to do a series of experiments that target some so-called endpoint, like reproduction in a fixed amount of time, growth rate over a predetermined period, and so on. Expose an organism kept under very idealized conditions to these different treatments and get a plot of some kind of response against a dose. The curve commonly has the shape that's described there, and there is a number in there called the no effect concentration, if such a thing exists, where which is characterized as the level at which there is well, as the name says, the level at which there is no effect, and a level called the EC50, the concentration at which the response is reduced by 50%, or whatever the response might be. And quite often, this picture can be drawn in different ways. It's sometimes written, sometimes plotted upside down, and so on. The key thing is that these metrics depend on, in particular, the EC50, but both of them actually depend on the details of the experiment. And if you're now going to regulate something or make decisions based on a measured EC50, even if you throw in a safety factor, you have to recognize that you're not measuring some piece of fundamental biology. You're actually getting at something that is very, very constrained by the details of the protocol. And that's why EPA and OECD and other organizations set aside very, very precise protocols for intercomparability of these types of measurement. And there was a very nice paper by a Dutch group led by Bas Kooijman in Amsterdam, and a graduate student who had amusingly had the name Bas in the same group. But Bas and Bas, if you like, did an experiment or looked at data from study, the sort of thing that is done regularly, growth and reproduction in C. elegans at different levels of exposure to a contaminant. And they considered two possible endpoints: the growth over a certain period of time and reproduction over a period of time. They used some of the standard methodology in order to estimate the EC50, and these graphs show you, well, this one, actually the EC50 for growth isn't there. This is the EC50 for reproduction. This is the EC10 and EC5, just the corresponding values for 5 and 10% for growth, and the broken lines are the values for reproduction. I don't want to go into the details. I want you to look at this picture and take on board the fact that you will come up with a very different number if the arbitrary rule said that you ran the previous experiment for 4 days, 6 days, 8 days, or 10 days. And if you want to now transfer this over into saying anything about what's going on into the environment, you're going to have to be very careful. And one way of doing it is to relate these studies back to what's actually potentially going on with this particular animal, rather than just worrying about a metric. And this is the natural lead-in to the approach that we use in perhaps 60% of the work we're doing here, and that I'm going to focus on this morning because it gives us the generic model that is the focus of this presentation. It's a set of models called, or family of models called dynamic energy budget models, and one particular group of these models was developed by a Dutch theorist, Bas Kooijman, and he certainly pioneered the application of this methodology to ecotoxicology. And in fact, he also collaborated with Tom Hallam at various points, and was a lecturer at one of the Trieste meetings that Lou talked about, one of the many connections I made that was impacted by that. There are other approaches than Bas's approach to the things I'm going to talk about. Some of them will carry over, some of them won't, but I'm going to restrict myself within this presentation to that approach. And in a nutshell, what a dynamic energy budget model does is treats an organism as if it were a chemical reactor. And you can think of it in a broad brush way that an organism is a chemical reactor that has inputs, which are resources from the environment, performs functions, which are growth, development, reproduction, avoiding mortality, and so on. And through the execution of these functions, produces metabolic products, which can be nitrogenous waste, CO2, inorganic nitrogen forms, various other quantities. So it's an input-output system, and it's characterized by some dynamical system in here that is associated with these functions that we're interested in. DEB models have been written down for many different taxa. In the next slide, I'm going to illustrate what it looks like for an individual, I should have said animal, in the header there, so a heterotroph. The idea is that food is ingested by an animal, some fraction of it is assimilated and goes into a compartment that within this framework is given the name reserve. I could talk after the talk or some other point if somebody wants to get into the detail of these various concepts. The use of the term reserve here is slightly different from the traditional use, but you could think of it as entering some form of recently assimilated food inside the animal. Reserve can be mobilized for many purposes: to meet maintenance requirements, for growth, for juveniles to enhance development or the increase in maturity, and with adults for reproduction. And the basic idea of the dynamic energy budget model is that you have differential equations that describe either the amounts of material or the amounts of stored energy in these various compartments. They're related to the environment. If you associate some stoichiometry with all of these, you can get the rates of production of metabolic products that I talked about. But at the heart of it is a set of differential equations that characterize the dynamics of the organism. And I've written, I've written 4 of them down here. And there are a couple of others for, I got 2 slides out of sequence here, that's what I've done. But all that matters is they are balance equations. And what we work with are actually a system in principle with around 6 state variables. The first 4 I've touched on. There's the amount of reserve in the system at any given time. There's the amount of structural biomass, which was the bottom left in my previous picture. There's the total amount of energy or resource that's been assigned to reproduction. There is this elusive concept called maturity that I can talk about, but you could think of it as a measure of physiological age, physiological complexity. But then on top of these that I've illustrated, there are 2 others that relate to mortality and are potentially quite important for ecotoxicological applications. And one is what Kuyman and collaborators have called damage-inducing compounds. And these you can think of as being exemplified by reactive oxygen species that are produced through metabolism and are both essential for some biological functions, but in significant quantity can cause different types of damage, ranging from DNA damage to cell membrane damage and so on. And then there are different measures of damage that may well be different depending on the organism and the situation you're dealing with. You might want to note with these choice of variables that they're actually pretty abstract. Although they're biology-based, they could be reinterpreted for anything from a bacteria to a blue whale, depending on how we're considering. So there is the possibility for some common, at least conceptual framework for thinking about a broad range of organisms. In the so-called standard form of Kuyman's model, Depending what you count as a parameter, there's around a dozen parameters. One nice feature is that a number of them are expected to be broadly invariant, and I mean by that probably not varying by a factor of more than 10 or 20 in value across taxa, whereas others scale predictably with size. And for many applications, and I'll demonstrate this in the talk, we can actually get rid of some of the variables, so we can deal with an organism as as being a dynamical system with a dimension lower than 6. So although this is the framework we're working with, it can be simpler. The whole gory story is set out in the book by Bas Kooijman, the third version of which came out 3 years ago, and if you're seriously interested in its supporting materials, it's the place to go. It's pretty heavy going, and the writing is a bit obscure in places. If you are interested in getting at the guts of the story, the key equations without without too many distractions, there is a very nice paper by Tanya Souza and 3 others. Bas is an author in this, but Tanya, who writes just beautifully, and who is at Lisbon, is the lead author of this paper, which appeared 3 years ago, that essentially tells the story without clutter. The payoff from that is that you don't get all the biological background there, but it's very clear, and I like to recommend it as a starting point, especially for people with a mathematical perspective who want to see the equations and then think about it rather than go on a random walk that leads to the equations. So, like I said, there's a bunch of differential equations. I've put a few of them up, but all that matters for the purposes of this talk is that there are a set of differential equations that describe the system. So what has this got to do with ecotoxicology? Well, from a physiological perspective, if we're thinking especially of sublethal effects, what do contaminants do? They potentially reduce the rate at which an animal feeds. They can increase maintenance costs because there's a need for detoxification, which requires energy. Directly or indirectly, they can slow growth. Directly or indirectly, they can reduce reproduction. By indirectly, I mean, for instance, that reproduction could be reduced just because there's reduced feeding and higher maintenance, or reproduction could be reduced because of endocrine disruption and be a direct effect. And of course, they can lead to increased mortality. The first 4 of these certainly, and in some circumstances the last, relate directly to energy budgets. So this, I would argue, is a reasonable way of approaching the challenge of modeling in ecotoxicology. And how that relates back to Bas Kooijman's model is that you take the picture that I've just seen before, and then each of these mechanisms that I've talked about relates to one or more of the flows in this picture. So if maintenance increases, we want to imagine, for instance, a parameter associated with this process changing. If feeding rate is reduced, we want to think of parameters there, a changing in value, and so on, and so on. And I can sharpen that up a bit, because really what underpins all the work we've done is that exposure to toxicant changes the values of the parameters in our DEB model. And this is why I call it biology-based theory, because the parameters represent biological processes that are going on within the organism. They're not directly related to protocols defined in some arbitrary set of experiments. But I need to elaborate just a little on what we mean by exposure in this context, because these rates may respond in a few cases to, for instance, the concentration of a contaminant in the environment But in many cases, an organism's response relates to the amount of a contaminant that's actually inside the animal or inside the cell. You can handle that within the DEB framework by simply coupling some kind of model that describes bioaccumulation, that's absorption and release of the contaminant or absorption, transformation and contaminant, to the DEB model. And you can relate the effect on the model parameters to either the internal or the external concentration. Again, you can get into even more detail. You can argue that there is some segregation of the contaminant between the structure and the reserve. In fact, that reminds me of yet another paper by Tom Hallam, which certainly wins the prize for the best title of a paper in this area, which is Survival of the Fattest, where he talks about a separation between fats and lipids and the rest of the body. In other words, we're still really working within the sort of dose-response area, but we have a much sharper idea of what we mean by dose and a much sharper idea of what we mean by response. If you want an introduction to this type of modeling, and in some of the literature, what I called bioaccumulation and toxic action models are called toxicodynamic and toxicokinetic models. I strongly recommend this little ebook by Chanling Yager. It's free, it's on the web, it uses a dynamic energy budget-based approach, but it contains a beautiful, almost equation-free description of the DEB models, and then it has a technical appendix which is longer than the book with all the equations in it. But Chanling belongs to another group, to the same group that I was suggesting with Tanya Souza, somebody who writes just beautifully, clearly, and simply about this stuff, and the book is free. It's a great download. So with that kind of overview, I now want to demonstrate that this approach can help us in this a larger enterprise which is trying to understand something about the effects of engineered nanoparticles in the environment. And I want to develop three themes. I want to show you the limited progress we've made in relating suborganismal observations to observations at higher levels of biological organization. So that's in terms of my description of the types of data. That's from the stuff that you can get lots of, but whose biological relevance is not so clear, through to some relatively simple population processes, in this case phytoplankton growth rates. I want to show how suborganismal information can also be used in model selection and supplement some statistical approaches. I want to pick up on this idea of metabolic products, because the very fact that organisms release metabolic products into the environment in turn introduces a potential mechanism for feedback. And I'll do that through another example. And finally, I'm going to say something very brief about individual to population projections. And I think it's fair to say that's the kind of question that people like me and Buzz Koyman and Tom Hallam and others were asking 20 years ago. This is just an application. This is just an application of the sort of ideas that have been around for a long time. Some of this stuff is conceptually much, much newer. And the hope is that these examples will give you some kind of feel for how models can help bring some more unified perspective here. So let me start by giving an example where there is information on suborganismal observations, which in this case we want to compare with higher levels of organization. And the next slide actually shows a some data taken from a large number of graphs in a paper on phytoplankton growth. And it's very short-term studies of phytoplankton growth, but again, a type that's commonly done where they're still in the exponential growth phase. So phytoplankton are growing in the exponential growth phase. In this paper, they were exposed to two metal oxides, TiO2 and zinc oxide, and there's a whole bunch of graphs in this and a subsequent paper. I've just picked two out relating to effects of zinc oxide on two different phytoplankton species, and what you see is unsurprising. It's just that the population growth rate is lower with the higher level of contaminants. You can take Kuyman's model and strip just about all the variables out of it until you're left with a structural biomass and reserve, and you get a much smaller number of parameters, only 3 that matter. And so we finish up if X here represented the phytoplankton population growth rate, and this is just the intrinsic growth rate. This relates to a bunch of DEB parameters, which I actually should have got rid of that. Which are they're just model parameters, and the key thing is that we assume that each of these are functions, in this case just of the concentration in the environment. It's maximally simple, and there are 2 parameters. There's a parameter that's related to not quite the EC50. There is a no effect concentration parameter, and you can just take these curves and you fit that and you come up with a no effect concentration. The kind of thing people have been doing with these models and similar models for at least 20 years. But you come up with a number which is for the no effect concentration. There's then a set of studies on the same species of phytoplankton, but done in a different lab, on the concentration of reactive oxygen species within the cells, on mitochondria. Actually, that is an error. It is mitochondrial membrane potential here. That's okay, what's there? Another a measure of damaged membrane that is associated with cell death, and so on. And then we take one of these. This is just one of the graphs from the previous picture for Isochrysis galbana. We estimate the no effect concentration, and we get a value of a bit over 200 ppb. And when you put that on the graphs there, we're in. This one is the nice clean one. The other ones aren't quite so clean. But what you see is that these suborganismal metrics are suggesting that something significant is kicking in at the same level that a piece of undergraduate statistical model fitting lets you get out the previous model. It's an encouraging example for the people who like to advocate the use of these molecular techniques. It's a nice example where everything works out rather like you hope they would work out, that if you were to use this to estimate the level at which you got a substantially enhanced reactive oxygen species, you'd be about right. The variation in the other metrics is actually not too surprising, because if you think of the sequence of causality that I described, I was thinking of these guys as being damage-inducing compounds and these top 2 graphs as measures of damage. The one will go up and then the other may or may not go up with it, and I'll return to that at the very end of the talk. But this is a simplistic example that says that at least it's not nonsense to try and relate the information at these lower levels of organization. The next example, which relates to model selection, involves a much more sophisticated set of experiments and a much longer study. Collaborator Trish Holden, a professor in our School of Environmental Science and Management, and members of her lab, did a series of studies of the response of bacterial populations to exposure to cadmium-selenium quantum dots, which are used in electronic applications. And the questions that we asked when we got these data was first a specialist question that what I would say the nanotechnology world wanted to know the answer to, which was, is there mechanistically a separate effect of the nanoparticle, or is it just a way of delivering cadmium to a cell? And we all know that cadmium is toxic. And so this, if you like, was a specialist question. We, as a modeling group, also had what I would call the more general question: could we use this, some suborganismal information in this system, to infer something about the mechanism of toxic action? And our approach here was to use the dynamic energy budget model to characterize the differences in the growth response when exposed to ionic cadmium delivered as, I think it was cadmium acetate, and the same amount of cadmium delivered as quantum dots. And I'm going to try and summarize the approach and the main findings here without getting too much into the weeds. And it's I may be trying to make an unsatisfactory compromise of too much information for some purposes and too little information, because the actual story is spread over 2 papers. But the basic idea starts by identifying all the many ways in which quantum dots could impact the organism, and it's moderately complicated. You introduce the quantum dots. Some of them will dissolve. The bacteria could be exposed to quantum dots, or they could be exposed to ionic cadmium. There's various mechanisms of quantum dot toxicity and cadmium toxicity, which lets us predict damage-inducing compounds. We want to relate these to measured values, the suborganismal value of ROS, and we then use that to predict population growth and compare it with what's observed. All that I think you need to retain from this is that the bottom row in this picture represents quantum dots, the top represents ionic cadmium. And in what we thought would be a training wheels type of exercise that would take maybe 6 to 8 weeks for the postdoc that was starting on the project, the first thing we thought we would do would be, well, fit the data on cadmium, on dissolved cadmium. It's a simpler system, and we've done things like it before. So we got rid of everything to do with the quantum dots. And 18 months later, we found out that a lot of our problems were that there was actually some additional feedback to the environment by these cells other than just utilization of substrate. In the paper, we call it environmental degradation, which, like many of these terms, just says we haven't a clue what it is, but we gave it that name. One possibility is oxygen depletion in the wells, in the 96 well plates used in the study, but there are others. And that story is sitting there in the +1 paper, of which Tint Planschek is the lead author. The original paper from Trish Holden's group is in the Priester et al. Paper that is listed at the top there. Eventually, however, we were able to come up with a model that gave a good description of the response to cadmium. The primary effects involved a toxic impact on resource assimilation and a mortality effect, and we were able to choose that model because we made the assumption that we could identify the damage-inducing compounds in Bas-Kooijmans theory with the broad spectrum measurement of ROS that the people in this lab did. And there is a question there. So if you like, when I say we have a model that says that these are the mechanisms conditional on us correctly interpreting or correctly making the link between one rather abstract model variable and one direct measurement. So to that extent, the modeling was facilitated by the connection with the ROS data. But we took that approach a little bit further. We then I've jumped ahead of myself. We were working with this model that had the variables, the reproduction and maturity variables were irrelevant for bacteria. We did have this damage-inducing compounds were related to ROS. We had a new variable here in the environment, and we had a new variable for the quantum dots in the bacteria that actually, again, well, this time related to the very early phase of a development, a phase of a population growth. So we now had a monster. We had a model with a lot of different possible effects. For remember, we didn't know what the quantum dots did. And we followed the approach that many, many modelers follow in a situation like that. We put the mechanisms one at a time in and in combination, and we did goodness of fit tests on all of them and looked at things like residual sum of squares and for some of them AIC, and we asked ourselves what were the best fitting models. And I think we could say we could separate, if you think of models as good, bad, and ugly, we could identify the ugly and we could get rid of the ugly models by by that approach. But really, all the others required in giving your data, the precise values of your data, an incredible degree of confidence they didn't deserve if you were going to really believe these very small differences in either AIC or residual sum of squares. So we had to come back to these ROS measurements. And let me just put all of these up. These are actually all the different mechanisms that we were thinking about, and these could relate to what was going on in the model. When we came back to the ROS measurements, we came up with a metric which was the ratio of the concentration of the ROS when the bacteria were exposed to quantum dots with the ROS level when they were exposed to the same amount of cadmium. In other words, it was a measure of the strength of the toxic effect. And we only had 4 data points, of which 2 were zeros, and they're on here. But now you look at your model predictions here, and you come up with 3. And I leave it to you to argue how much credibility you give to this, but if you now actually believe our identification of the state variable with the measured quantity, recognizing that this is on a log scale, there is actually an argument for saying that all things being equal, we have separated these models out in a way that is different from that is arguably more convincing than a difference of 0.2 in an AIC value or something like that. So I don't want to overstate the result, but we potentially are in the situation where we have a general conclusion, and I think this is robust. I don't believe that by simply looking at lots more growth trajectories, we would have gotten ourselves a better ability to discriminate among potential mechanisms. The model had too much detail, and the data was too similar. It was too much all of one type. And we have one candidate way of discriminating in here, and whether or not you like that method, that particular discrimination, I think it's fair to say that something like this is the way we're going to have to go in future work. So that's about relating the suborganismal to the organismal. The second piece of modeling that I want to demonstrate relates to this feature of DEB models that they describe the production of metabolic products. And my starting point here is a set of experiments on phytoplankton growth, this time a freshwater phytoplankton, Chlamydomonas, performed by Louise Stevenson, a grad student in our department, working with Ed Macaulay and me. And Louise got a surprising result. she was doing a series of experiments and discovered this almost by accident. She was running a series of batch cultures in Chlamydomonas in order for us to parameterize a fairly routine model. She exposed silver nanoparticle, or introduced silver nanoparticles at a given level to some cultures which had a relatively low concentration, some cultures which were about a week old, and some cultures which were about 2 weeks old, and then followed the phytoplankton populations, or chlorophyll A, which is, I think, a reasonable surrogate in this case. With the new culture, the population bombed. It was just super toxic. With the older cultures, there was a long period of hanging on, followed eventually by a toxicity. There was then a series of auxiliary observations, some of which I've summarized here. Silver nanoparticles she was working with have a fairly slow dissolution rate. The critical one was, if she then took some water from some of these cultures, removed the organic, well, essentially took them from water that had lots of dissolved organic carbon and put the old cultures into fresh media, then it was toxic again. In other words, the cause could be the cells had changed, or the cause could be the environment has changed. This and a couple of other studies argued that the environment had changed, and then a quantitative one that dissolved organic carbon compound level was shooting up. So this leads us to a hypothesis that says the algae are excreting organic compounds. These interact with the nanoparticles and reduce the toxicity. So if we're going to understand, if this is right, if we're going to understand the dynamics of populations exposed to contaminants, then we have to consider a feedback where potentially one component of the system is producing product, releasing, excreting stuff that in turn influences the toxicity. It's a different feedback from the one that almost every study I know that uses ecotox models for populations has considered. And we've put together a model based on this core idea of the dynamic energy budget theory, which was that the cell in this case is a machine that turns resources into metabolic products. If the model is right, then if that picture is right, and if our representation of the organism, which at its simplest level is just two variables here again, reserve and structure, because it's unicellular, then any of the metabolic rates, you can prove this quite quickly, can actually be written as a linear combination of the total biomass of the algal population plus its rate of change, where these are just arbitrary multipliers. And that let us get rid of the need to have a detailed algal model, because we then used the control data to get ourselves these and fitted it to the data on DOC production. We then put together a model that had more detail, but still minimal detail about the interaction of the DOC with the particles and with dissolved silver. So you had silver nanoparticles, silver ions, inactivated silver ions, inactivated nanoparticles. The rate of inactivation was accelerated by DOC. The bioavailable parts had a negative effect on phytoplankton. It's a relatively simple model with positive and negative feedbacks. There's half a dozen parameters to be fitted, and when you fit them, you get a very nice fit. The previous curve I showed with Louise's data just had lines joining the points. You can get a very nice fit to the data. That itself is not particularly convincing because we had 6 parameters, but the parameters come out with sensible values, and in particular the dissolution rate that is estimated here comes out in the same ballpark as is found. So I think that set of experiments shows the importance of a metabolic products for subsequent ecotoxicological modeling, and at least gives us some hope that simple representations of what's going on in the environment will suffice for describing it. Just to try and convince you a bit further that this matters, we did an experiment with my favorite animal, Daphnia pulex, and the animals were treated to an absolutely standard 48-hour acute toxicity test. That's just a question of expose them to something nasty and see how many of them can live for 48 hours. In fresh medium, the red curves represent the response to increasing exposure. If the nanoparticles were added to medium from the later stage batch cultures that were full of this dissolved carbon, none of the animals died. So there is potential protection in here. And again, we're getting information on the importance of feedbacks. The previous example, the quantum dot example, required us to think about an extra term that we called environmental degradation. Here we're talking about the physiology actually leading to some protection. My final example, which is very brief, I'm just going to, because I think it is an approach with which many people are familiar, but it still gave a result which was a bit surprising, which came as a surprise to a number of our collaborators in the center, and it relates to individual to population projection. And the idea here is that you can take a dynamic energy budget model, you can run it through time for organisms exposed to a particular environment, you can compute the fecundity as a function of age, you can either compute or make assumptions about survival to an age, and you get the quantity R, sometimes called R0, that is proportional to the lifetime reproductive output of the animal. The interest here was in experiments on marine mussels, which live for several years. The experiments, which ran for a couple of months, were performed by Shannon Hanna, another UCSB grad student, where he did a large number of what I would call routine, and I know this is easy for a theorist to say, but a relatively straightforward standard measurements on individuals at 2 different food levels. He and Eric Muller, a professional researcher in my lab, worked together to fit the parameters of a DEB model. Here we actually got a big benefit from the use of what I'm calling a generic model and a model that other people have used, because there actually wasn't enough data in Shannon's experiments to fit all the parameters. But about 5 different groups in Europe and 1 in the US have used Bas Kooijman's model in studies of mussels, and all the information is tabulated in a website nicely called Add My Pet in Kooijman's webpage at Amsterdam, or is published, so we could get a good idea of the other parameters. And that in turn let us estimate what is the effect on lifetime reproduction of exposure to these particular particles. The results are summarized in, actually, no, that's the experimental paper. The results are summarized in this paper, which is currently in review. I don't want to go into the details of the color coding in this picture. I just want you to think that this curve here is a reasonable approximation to the EC50 for lifetime reproduction, and to note that in these units it has a value like between 0.2 and 0.3. The EC50s for the individual physiological rates, when they were measured in the lab, were many times this. They were they were greater than 1, and for feeding, for instance, there was a value of around 1.5. And what this tells you is that if you had based an evaluation of the toxicity on the results only of these short-term physiological experiments, you would have been wrong in the dangerous direction, That's to say, you would have reckoned that that the problem was kicking in at levels, kicking in a big way at levels above 1.5, and here you're saying it's kicking in at levels about 8 times smaller than that. The model lets you understand why. The reason is relatively straightforward, and again, the reason I'm doing this one fairly quickly is that this is something that's been recognized for a long time in other contexts, and that is that if feeding rates are reduced and metabolic rates are increased, there is a direct effect on reproduction just because there's less food coming into an adult, but there is also an effect because the adult is smaller, the final size is smaller. So there's an effect associated, a direct effect on reproduction, and there is an indirect effect on reproduction associated with reduced growth, and these combine to give you a more serious effect. And that is something, again, that I think many theorists are well aware of, but it has not spread out into the wider ecotox community, and is therefore a reasonably useful take-home message. So I've tried in this to summarize, well, to introduce you to the ideas of our approach to using DEB models to interpret results and suggest new studies in this broad range of environmental nanomaterials. I've focused on 3 particular ways that we can do it. One is to learn more about ways of using data on suborganismal processes in order to test different models. The second is to recognize the importance of metabolic products, because I, at least as somebody who's worked for a quarter of a century in DEB models, has largely, up till a couple of years ago, sort of ignored and not been bothered about this business of metabolic products, and I was wrong, and that is coming very, very clear. And finally, there is this question of individual to population projection, and that actually could have been the theme of a talk I could have given 15 years ago. You know, that's been around. So, try 500, Lou. Yes, a long time back. So in that context, what are we doing next? We're particularly aware of this point that I've touched on at various points, which is that a lot of our work rested on having an idea relating to oxidative stress, which is the mechanism of toxicity in a lot of these cases, and the production of something I'll call damage in air quotes, and damage can be different things. We are actually currently, that's Eric Muller, Tin Planchek, and myself, focusing on getting rather general models of the different feedbacks that are possible there, and you can actually lift a lot of this out the systems biology literature and reinterpret. I have one paper, for instance, that has, I think it's 50-some different feedback schemes all tabulated and their properties listed. The challenge is then to read it, but that is we're definitely focusing on that. We are, as you'll have gathered from what I've been saying, increasing our emphasis on understanding these material fluxes, and that is going to be particularly important for the work of Louise Stevenson, who is starting a new set of fairly ambitious experiments on phytoplankton-zooplankton interactions, which, before Lou says it, I'll say is also something I could have talked about a long time ago, but now with nanoparticles. And not yet started, but downstream there are a set of estrogen mesocosms being constructed at Bodega Marine Lab, which is north of San Francisco and linked to UC Davis, and we're collaborating with the people on that project, where they'll be using copper nanoparticles of a wide range of types and looking at the effects on small artificial mesocosms with different levels of salinity. And that's a sort of very broad tour, and I hope you see what I mean by saying there's nothing grandiose in any of this, but there's a set of projects in which a systematic mathematical modeling approach gives incremental progress. Thank you.